So, welcome. Thanks for everybody for being here. We are going to have a two-part series. Tonight is part one of talking about our Roots to Shoots trial that we did with RhizeBio. So we're lucky to have Josh Toll and Keenan Gerhardt here with us tonight. We'll start with a little introduction about who we are for the new folks that are on the call tonight. Thank you again. My name is Camille Newsom. I'm the Director of Grower Support here at EnSoil Algae. And we have a handful of folks from our team here tonight, Christopher and Meredith and Yazzie. So we'll all be on the call to answer any questions that anybody has about EnSoil specifically. So with that, we're going to do a little introduction, and then we will get started with the RhizeBio. So we at Enlightened Soil Core, our mission here is to empower farmers and land stewards in the effective and profitable implementation of a regenerative agriculture system. We do that with partners like Josh and Keenan. So that's why we're doing this webinar and a lot of the other webinars that are sort of in this Stacked Solutions series more broadly. As a company, we're committed to soil health, education, and profitability of farmers and land stewards. We're all guided by our connection to the earth and the interdependence of life. We're inspired by farmers, by nature's design, and by human innovation. And we all believe in this ecological systems approach to food sourcing that is economical and nourishing for people and the planet. We grow a live cell algae product. It's a green microalgae freshwater called Chlorella vulgaris. And I think you guys, okay, you can still see the slide. Sorry, something's weird going on my screen. So you can see the cells in the upper right-hand corner. This is a liquid concentrate, and it's used in many agriculture systems across the country and actually internationally as well. So this living algae is applied through any irrigation system. It is tank mix compatible in all growing systems. We grow it in the US, and it's used to improve things like soil functionality and plant functionality. In addition to the crop production, we do see yield increases, stress adaptation increases, and really a lot of what the algae does you're going to hear about from Josh and Keenan tonight as we sort of review this trial that we did with them this past season. So with that, Meredith, do you want to tell a little bit about how we know RhizeBio and the story behind that?
Yeah, thank you for joining us, everyone. I think it was around 2022, Tucker met Josh and Doug at the Southern Farm Show. We started talking to them and really learning about their specific type of genomics technology, and really loved what we were seeing there. We are always seeking, deeper knowledge of the system and how the algae is interacting in the system, and we really could see that we could partner up with them to do that. Yeah, and we started, really traveling with them and, growing our relationship personally and professionally. It's really important to have integrity in the technology. We saw that so much in their team, and we also all have a shared sense of humor. And so it's been a lot of fun to grow together, and we've really enjoyed getting to know Keenan as he's joined the team too. They have just really, this team has helped deepen our own knowledge of the modes of action and mechanics happening, the mechanisms happening there. So we're just grateful for this relationship. And we utilize all of these tools to help teach our growers while we're transitioning, how to better understand and manage systems. So this is just one of those amazing tools that we're utilizing there too.
Awesome. Thanks, Mare. So with that, I will introduce both of you, and then we will get started, and y'all can share your presentation whenever you're ready. So Keenan Gerhardt has a degree in environmental engineering. He began his career with Superfund soil reclamation work before transitioning through continuing education and on-the-job experience into agriculture, specializing in the soil microbiome. He's thrilled to be a part of the team at RhizeBio and to contribute to furthering our understanding of how the soil microbiome can unlock yield, plant health, and profitability for farmers. He brings a driving passion for the natural world and a strong desire to leverage soil and biological data to help farmers make informed decisions that drive innovation, protect their bottom line, and promote healthier soils, healthier food, and a healthier environment. And Josh Toll is the co-founder and COO of RhizeBio, an ag tech company that really bridges the gap between soil and crop health through DNA sequencing and biological insights. He works alongside farmers and input manufacturers to unlock a deeper understanding of the rhizophagy cycle, how soil microbes support crop health, helping them to apply biological data to improve field performance. So Josh and Keenan, welcome. We're so glad you're with us, and take it away.
Thanks for having us.
Yeah, thank you so much. What a great introduction, and just so thankful. It's been a pleasure working with both y'all, the EnSoil team, and also all of the farmers have been wonderful. With all the farmers that we did these trials with, we actually met and went over the results and talked through every line item just to kind of understand what exactly was happening in their specific system. Every soil is different, every crop is different. And so maybe just on a high level, I'd love to just talk about RhizeBio, what we do, why we're different, our approach to metagenomics. It's a big word, it's a complicated system, but we're doing our best to make things more simple and really get to the root cause of health, genetic potential for crops to produce life. And so we my father and I actually started this company back in 2022. He has over 30 years of clinical diagnostic experience in healthcare. So using these systems biology tools to see if somebody is predisposed to Alzheimer's disease, or looking at the human gut microbiome to find really unique patterns that will help inform on how folks can improve their health. And so we took his background as a mad scientist in healthcare, and we're applying it to soil and to roots. And so really thankful and for the opportunity to work with EnSoil. Can y'all see my screen here?
Yeah, you're good.
Okay, awesome.
And if at any point y'all want to ask questions, just throw them in the chat, and Keenan and Josh and us will answer.
Yes. So at RhizeBio, we specialize in the root system, the rhizosphere. This is the 1 millimeter of root-associated soil that's closely bound to the roots. And what we do with that 1 millimeter root-associated soil is we will actually sequence the DNA, all the billions of microbes that are surrounding that root system. We're looking at who's there, what they're doing, whether or not they're cycling nitrogen or phosphorus, potassium, if there's any pathogens or stressors in that environment. We're also looking for plant growth promoters like phytohormones and things of that nature. We all know that the gut microbiome is so critical to human health, and the same applies for crops. Crops have guts too. And so we are experts in gut health for crops, to put it simply. Now, one thing that just want to kind of give you an understanding of why we focus on the rhizosphere, which is that 1 millimeter around the roots, is because when you pull a sample from a bulk soil, 90 to 95% of those microbial populations are not directly interacting with the crop. And so we set out with our technology, with this really advanced technology, looking at all of these microbes. It's a lot of noise. And so we want to simplify it, get down to the data that's really important for the grower. And so we landed on the root system. And to kind of take that a little bit further, in the human gut for babies, specifically infants, it's really important that they have a healthy gut microbiome. And the same applies for the crops. So that first 1 to 2 week of propagation is really critical for future health. So we do a lot of testing at that growth stage as well. But we also do, we have this Roots to Shoots program wherein we are going out in the field into a treated area or an untreated area, collecting several crops from that area, shaking off the root-associated soil, goes into a bucket. Doesn't have to be as fancy and cool as my bucket, But we would take that and send it out to Region Ag Lab to run a Haney analysis. So we're looking at organic matter, water extractable carbon, all of the pH and all of that good stuff. And then in addition to that, we will collect the root system. Roots will be sent to our lab. We will process the roots through DNA extraction and sequencing, and then we will also run tissue analysis over through Regen as well. And what we found a really awesome, interesting relationship between the soil that was being ran on a Haney and the root system and the tissue. And what that trend that we saw is that if the crop is cycling nutrients in the roots, in the microbiome of the roots, then we're going to see elevated nutrient levels in the tissue, and there's actually going to be a reduction in the available nutrients in the soil. So this is a way for us to connect the dots between the roots and the shoots. This is, and I called Liz, I know Liz is on the call. I called Russell, he says, all right, if I share this, but this is basically AgWise, and they have this program called foliar scripts. Many of y'all may be familiar with it, but this is kind of a graph of when nutrients are in peak demand for a corn crop. And this is something that we like to refer to whenever we're talking about nutrient cycling in the rhizosphere. We collect a majority of our samples at V4 to V6 when peak nitrogen uptake is occurring in the root system. We like to see that specific growth stage to kind of hone in and focus on nitrogen. But you also have some of the later stages, which we'll get into, silking V10 to V12, where we're seeing potassium and phosphorus levels improve. But this kind of gives us an idea of when we should expect certain nutrient pathways to be turned on in the crop. And I'm actually going to hand it off to Keenan here. This is in today's review, we did several different crops, anywhere from corn to soybean to milo, and wanted to just kind of highlight what we saw in corn today. And keep in mind, as we're going through these different reports, it's a lot of data, but they're separated into and kind of bend into two different growth stages, V4 to V6. I'm sorry, 3 growth stages, V4 to V6, V7 to V9, and then V10 to V11, I believe. And
10 to 12, and then reproductive silking stage.
Yep, exactly. So what you're seeing here, this is an overview. A lot of data goes into each one of these different metrics. On a high level, we have kind of like the health overview, and then we get down into the carbon, nitrogen, phosphorus, all of that good stuff. But on the left, what you see there is untreated. So that's coming from the replicated strip that does not have EnSoil applied. In the middle, that treated section has EnSoil applied. And then on the right, we have the percent change. And so roots to shoots, you see the Haney analysis, the root metagenomic analysis, and then the tissue analysis. I'm going to let Keenan take it from here.
Yeah, thanks, Josh. As Josh mentioned, he kind of gave us a run through. So we have both our treated and untreated here. And for both sets, we'll have a set of Haney data, which I think most people are probably familiar with, and a set of tissue data that most people probably have at least some experience looking at. And so right in between, we sandwich our metagenomic data. And like Josh mentioned, we kind of see that as sort of the gatekeeper, right? Sort of the pathway to go from the soil into the plant so that you can make sure that the nutrients you're putting down are being delivered effectively. And this both helps, inform on what's improving nutrient levels in the crop, and as well as, what kind of nutrients you might be lacking in the soil, or what kind of biological inputs might be useful to help and unlock the potential of the soil to actually help cycle those nutrients, because it's kind of an underestimated portion of a lot of row cropping systems is what the biology can play. And especially in, as Josh mentioned, that rhizosphere, the soil that really binds onto the roots, there's It's really where the action is, and the plant is putting out exudates, it's putting out sugars that actually feed the soil microbes that the plant wants to have in its root system. And in exchange, those bacteria and fungi, they can actually perform some functions that the plant can't perform for itself, right? They can transform nitrogen, they can solubilize phosphorus. Phosphorus is a big one. They say that there's enough phosphorus in most farm soils to satisfy the plant needs, but it's bound up, especially in clayey or iron-rich soils. You have a lot of trouble getting that phosphorus in ion forms that the plant can take it up. And so that's a way that the microbiome through the kind of rhizophagy cycle can serve the crop and at the end of the day serve the farmer and serve the bottom line. So this is a cool actually example of the trial, because as I go through, you'll be able to see a couple of different instances where I guess first I'll kind of run through and talk about what some of these pathways are that we're looking at, what some of the microbial activity that we're going to be looking for. And then we can kind of trace different nutrients from the soil through the microbiome and into the tissue. And hopefully, like Josh mentioned, we'll see an increase in the activity in the microbiome. We'll see an increase in tissue nutrients. And then corresponding, we'll see actually usually a slight decrease in some of the soil nutrients because they're moving into the plant. But sometimes you will see increases in those soil nutrients as well, because the system can actually kind of bank nutrients, right? It can store up a little nitrogen, or it can store up a little calcium or sulfur, or whatever that is in the soil for later use, or for just the kind of health of the whole system. So first and foremost, I'll kind of start I'll just do a quick rundown going down. So community structure, we basically look at who's there, right? How many different types of microbes are there? Are they really competitive, or is one or two types really crowding everybody out? And so we will give a score based on that. I do want to point out that you'll see these middle numbers, they're percentages. And what that really means is we're actually comparing this crop against all the other crops that we've studied so far. So in this case, we see for community structure, we have a 44%. That means that this soil scored better than 44% of the other soils we see. Or if we have a 75, that means we had better respiration in this soil than in 75% of the other soils we tested. So they're all kind of relative measures, but just to kind of give you a sense of what we're, all these numbers get a little scary, but that's why we heat map it a little too. We add some colors, so at least you can kind of get a good sense just looking at it. Respiration, obviously oxygen is very important. So we look at how much oxygen is available. Most of the microbial activity that we really want to see is going to require some oxygen. And so that's a good indicator of crop health. Environmental stressors, we can look at, different, I think in this case, really low oxygen environments, that whether it be from flooding or, a compaction or silt layer in the soil, we can actually see that the bacteria and fungi, they change, and we can see that response to those kind of stressors. And then we're going to look at all the kind of major nutrients. We're going to look at a couple different ways that carbon moves, a couple different ways that nitrogen moves within the soil, and then all of our phosphorus, potassium, sulfur, calcium, and iron. That's the markers that we report on right now. We're always looking to kind of keep adding to that list, but we feel really confident in our ability to understand how the bacteria and fungi and protozoa in the soil are affecting how those nutrients are taken up. And I did just want to point out a few quick things. I think Josh we got some cool slides where we can dive a little deeper. But overall, we boiled down these tissue numbers and all the numbers really into scores. And so just first and foremost, you can see that we saw on average about a 5% increase across all the markers in the biological activity that's driving that nutrient uptake and crop health. So that's a good sign, right? And we see an increase in the tissue. And that's not always an increase, right? There's a few interesting ones for, say, iron or aluminum. And a lot of those heavier metals, especially in low pH soils where they become really soluble, can actually become toxic to the plant or can inhibit the ability for the plant to take up other key nutrients. So sometimes we actually see a reduction in certain nutrients, in this case, that iron and aluminum, but it already has way more than it needs. No plant needs 400 ppm of iron in it for the most part. And so what that is going to do is going to allow the plant to take up more of the nutrients that it does need. And that's where we're going to see that increase in nitrogen, right? We get up above 5% nitrogen. a little bit of increase in phosphorus. We see more calcium, more sulfur, more manganese, a little more zinc. Almost across the board, we're seeing some of those pathways unlock, and we're seeing that the microbes are key in that, right? More microbial activity, more nutrients in the tissue. And that's one of the cool things to tie this back to EnSoil a little bit that we see is it can kind of help kickstart the biology that's there, right? The plant is going to have some relationship with some of these microbes in the soil that it's using all the time to try and get the key nutrients that it needs. And what it seems, right? And I think one of the things we've been able to help and work with the EnSoil team to help kind of learn how the product functions, it seems to really do a good job of sort of kickstarting, right? Giving a little boost to a lot of those microbes in it. It all tends to start with carbon. So we really like to see that increased and perfect. We like to see that increased carbon cycling, and you can see we go from kind of oranges and yellows into that high green. So we're pulling in a lot of carbon from the atmosphere and carbon fixation, and we're breaking down a lot of carbon. That's key too. So, you know, if you have cover crop litter or you know, corn stubble in the ground still, we can return those molecules, that carbon and nitrogen, more quickly back into the soil where it can be used. And we see a reduction in methanogenesis, right? Because that's going to lose carbon out of your soil, and factors into carbon credits. I think there's 45Z and stuff people are becoming more familiar with. So, and overall, we actually see more total organic carbon in the soil than we started, or than in the untreated sample. So you can kind of see that the way that microbiome can serve to build the health of the soil and the health of the crop, and make sure that the nutrients it needs are available when it needs it. So this was a really good story, I think, on this farm, and you can really see some of those carbon pathways unlock. And then obviously nitrogen is a big one. It looks like a lot of data, and it is. We have a couple different ways because the nitrogen cycle is so complicated, right? And one of the cool things that we see in this, we definitely see a lot more nodulating bacteria, which is interesting because this is a corn. It's not a soybean crop, but and we see actually more nitrification. So that's going to be moving from ammonium into nitrates, very plant available form. We already had really high N fixation. And so we don't see actually, we don't see a really big difference in the nitrogen fixation in this, but I guess I apologize. Yeah, but we do see an increase in the tissue nitrogen. And so overall, you can see the way that even just slight increases, slight changes, and I forgot to mention that organic nitrogen breakdown as well. We see an improvement, and that's huge because you can develop or deliver to the crop some of the best forms of nitrogen, which are going to be amino acids. Same as humans, plants want amino acids, and so that's really important. And you can see about a 0.4 of a point increase in the tissue nitrogen in this particular sample in response to some of those changes in the microbiome.
Yeah, and to add to that, Dan Jansky, he already had really good levels of nitrogen in the tissue. According to Region Ag Lab, they have like a sufficiency range based off of different growth stages of the crop. And so Dan already had a good amount of nitrogen in his system, but we still saw a 13% improvement in the available nitrogen for the crop at this growth stage, which I think is worth noting as well. And by the way, this is one farm of a number of different farms that we looked at, and so we'll kind of jump into the V4 to V6, combining all of those averages together for EnSoil for all the farms that we analyzed. But we wanted to use Dan Jansky's soil as an example because while we saw really great results in the roots, in acquisition of different nutrients, stabilizing some of those micronutrients that might be toxic for the crop, we saw a 12 bushel improvement against the untreated field. So everything seemed to be aligning with what we were seeing in the lab, and then also what Dan ended up seeing at the end of the season.
Hey, Josh, do you mind popping back 2 slides? We got a question about the nitrogen. That's actually a really good point. I kind of glossed over that. So one of the things that we like to be able to sort of combine in this report is to be able to see the amount of ammonium nitrate specifically in the soil, as opposed to, both total nitrogen overall and what's present in the different ion forms of nitrogen. And this one, we don't see huge departures because, again, already had really pretty high nitrogen fixation, pretty high nitrification. So there's pretty healthy nitrogen cycling already going on. But that is one thing that we can expect to see is sometimes you will say we have a ton of nitrate and we don't have much ammonium, right? And so we want to drive nitrogen fixation now. Or maybe we have a ton of ammonium, you're in maybe a soybean crop, and we need actually a little bit more nitrate. And so then you can kind of identify where the hold points are and where you can improve the system even within that nitrogen cycle to find the right forms of nitrogen to deliver to the crops. That's a really good question. Thanks, Kelly, for that, because I kind of skipped over that. But we do look at the different forms of nitrogen and how they relate to that microbial nitrogen cycling. So. You want me to speak on this one, Josh? Or?
Yeah, that would be wonderful. So this is 3 farms averaged across all of them. So if you want to talk to this as well, we don't have to get maybe pinpointing some of the major increases that we saw, and then I think I have additional slides on that. Oh, yeah. Then we go down to silking. So
yeah. So this is I think we're just looking here at we can look at how it works on one specific farm, right? And that can help us tell very specific stories about, like we were just saying, you're low on nitrates, or a phosphorus solubilization pathway is kind of blocked up, and you might be able to improve, phosphorus specific to this farm. But we're also with this trial, right? And we do this across different farms in different states. We're able to sort of average across those values and see how the product is performing overall. Right, and that helps us kind of inform. And I think this is, key for the EnSoil team to see what sort of changes you can expect overall in the crop. And one thing that I would note from this, you can see, and kudos to the EnSoil team, to a lot of, I suppose, the farmers on the call, these plants are already right in key sufficiency range almost across the board. Right, it's kind of hard to improve on some of these scores when you're already averaging 4.5% nitrogen and 0.44 pp or percent phosphorus, and we're looking really good across the board. And so one thing we actually look at is different nutrients can actually interfere with each other a little bit, and so sometimes actually less nutrient is better. Right. And you'll see that not just with the iron and aluminum that I mentioned earlier, some of those heavier metals that can really interfere, but even, some people are more familiar with calcium to magnesium ratio, right? Because that kind of base saturation, that ion, or the relationship between the positive ions in the soil can really determine what is able to be taken up and can allow more nitrogen and more phosphorus to be taken up, some of those anions, the negatively charged ions. And so it can be about balance. And that's one story that I think is overall can be told from these EnSoil trials is you see improvements in the soil microbiome overall, but you also see, I would say, more efficient uptake of nutrients. It's not always necessarily more. I think if you're already a little low on nutrients, you might be able to expect to see more. We do see that on specific farms, but overall, we like to see more efficient uptake. I don't want to spend too much time here, but yeah, you can hear, this is a really good example. Actually, I'll just kind of, you mind if I talk on this, Josh?
Oh yeah, go for it.
So this is really cool. This kind of goes back to that expecting to see maybe a slight actually decrease in what's available in the soil. But what's happening is the soil microbes are more active, and that's going to lead to increases in the tissue. So in this case, we had still pretty good phosphorus and potassium levels, but there's a little bit more room for improvement. And there's reasonable levels of phosphorus and potassium in the soil, not huge, but it's there. And then we can see that in the center column there, the RHIZE scores are going to increase by about 14 points and 11 points for phosphorus and potassium respectively. And you see that you're draining a little bit of those nutrients out of the soil pool and delivering it to the crop. And so this is exactly what we kind of like to see. This is a really good example of how we can identify not only the transport of nutrients, but how the microbiome, how the bacteria and fungi in the soil are improving that nutrient cycling potential, and that's serving the crop and leading to 16 and 17% increases in phosphorus and potassium, which is pretty great to see. I think anyone would be happy to see that from any treatment, whether that's a solid fertilizer, a liquid fertilizer, a biofertilizer. So overall, this is a really good story from this particular farm, and this was a late silking stage crop. So not quite during that peak growth stage, but obviously there's still a demand even later on in the reproductive stage.
And one thing that we've noticed, I did want to point out, when the crop's microbiome is performing really well, oftentimes in earlier growth stages, which was, if we just kind of hone in on this phosphorus marker right here, that 8.6%, it's beginning to build up available phosphorus, basically storing phosphorus, and also 35% improvement in potassium in the soil, and even for nitrogen, 17% improvement in the available nitrogen in the soil. It's not in peak demand right now, specifically for phosphorus and potassium at V4 to V6. Then once we get to silking stage, those pathways are turned on. It pulls the storage from that it was building up, and then dumps it directly into the crop. So something that we love to see, and a typical trend that we're seeing.
That's a really good point, Josh. This is just another look at that sort of pattern that we saw really well in that last sample, where you see slight decreases in what's available in the soil, translating through that increased microbial activity into increases in tissue nutrients. So soil starting on the left to the tissue on the right.
And then this is the silking summary report.
Once again, I guess, I don't see any.
Did we go? I think we already, no, we did not.
I thought we had one more slide in there, but let me check. I can just kind of run through this as well. So at silking stage, we also see, this is actually an interesting one. We saw a pretty strong response essentially across the board. There's only one nutrient there that didn't move further into sufficiency range, and that was manganese, which was already about dead center and only about a 3% change. But across the board on this, I think this was only 1 or 2 crops for our silking stage, and that's one of the things that we're excited for next year, is to get more farms involved so we can really kind of gain a better understanding and see how this pans out. But you're seeing increases in all your NPKs, better sulfur, actually a little bit lower calcium and iron, but those were a little bit high to begin with. Magnesium is I think that's a bit high, but it moves in the right direction. And you can see that 2% increase in the microbial populations, huge improvements in phosphorus and potassium cycling. And I think that's a really big one. That's one that, a lot of inputs get put down for phosphorus and potassium. And I think a lot of farmers don't realize how much the soil microbes can serve to improve the use of those nutrients, and how, efficiently, you're not losing so much to leaching or anything like that, and it's actually getting into the crop. So this is definitely a good story, even at that later reproductive stage, when we're not necessarily looking always for more nutrient uptake, but just making sure the crop has what it needs so it can divert so much of its energy up into those reproductive systems in the ears.
Yeah. One thing I like to, when talking about genetic potential or biological potential, 3 important points that I like to make are, the microbes, they need a home, right? In many cases, that's the root system. It also, pH plays a really significant role in that. It's interesting, EnSoil's products, consistently, if there was really high pH levels or really low pH levels, we always saw that moving towards sufficiency range and a healthy level. So having a home is really important. Having a food source is also incredibly important. We saw earlier on in the V4 to V6 that there was a lot of carbon that was being dumped into the soil through carbon fixation. This is microbial available carbon. This is water extractable carbon, which is immediately available for the crop. And I think going back to the carbon levels in the Haney analysis, there was about a 2% reduction in the available carbon. But then at later stages at silking, we see, okay, we see a 2.6% improvement. And so what I believe is happening is that, the crop is improving its metabolic function, and it's dropping carbon directly into the system through carbon fixation, through the atmosphere, through photosynthesis. And so you have your food, you have your home, and then we also need to breathe, right? So respiration. Respiration is really critical for crop health and yield and genetic potential. We see a 40% improvement in the crop's ability to breathe, whether that's, I believe specifically for the Haney analysis, that is CO2 production. And then in the RHIZE score in the root system, we're seeing higher oxygen production. So we have basically more of a better ability to breathe. So you cover those 3 points, and you've got a really good chance at producing really strong yield at the end of the season. So kind of just following these trends for the crop from these different growth stages. In the beginning, carbon fixation was through the roof. And then we also had a lot of nitrogen fixation and nitrification. And then we saw a reduction in methane production and denitrification at V7 through V9. And then at silking, we see a lot more phosphorus solubilization and potassium solubilization.
Just kind of a cool way of, sorry, I didn't mean to put it.
No,
yeah, you can just really see on the left, those big green bars are in carbon and nitrogen, right? And so at that stage, the plant is getting those essential nutrients, and it's really driving that carbon and nitrogen in the microbiome. And the phosphorus and potassium, still important, right? But that's going to kind of come later. And then you can see as it moves through the vegetative stages and into the reproductive stages, it's really going to, it has most of the carbon and nitrogen it needs, hopefully by this point, and it's going to really push for some of those other macro and micronutrients, and you see that phosphorus and potassium shoot up. So just, yeah, it's a little bit hard to see what's going on, but it is a really cool look at how the crop's needs change over time and how the microbiome responds. So go ahead, Josh.
Yeah, these are some of our main kind of findings that we saw in our root shoots, specifically on corn. 41% increase in soil phosphorus, 107% increase in soil potassium, 20% increase in soil nitrogen. And then at silking, we saw an increase in phosphorus in the plant tissue of 17%, 16% improvement in potassium in the tissue, 5% increase in the nitrogen in the tissue, and then these are the metagenomic pathways that we're looking at. 13% improvement in phosphorus cycling, 7% increase in potassium cycling, 5% increase in microbial nitrogen cycling. Wanted to jump into a program that we are doing alongside EnSoil. And this is our greenhouse program. So we just went through our infield protocols where we're pulling the crop, we're looking at fertility in the soil, we're looking at fertility in the tissue. This specific program is focused on what we believe to be the most important sample type in corn, soybean, really any crop, and that is that first 1 to 2 week of propagation. What's involved in this program is typical soil health protocols for collecting soil, 15 cores, 10 to 15 cores, 6 inches deep. We do require seed for this study, so it's important to know that every soil is different, every seed is different. Your seed actually recruits the native microbes or the microbes that are applied to the seed, And so we ask that you send your soil and seed. We'll actually propagate these in our greenhouse, and we will look at different nutrient cycling pathways, carbon, biodiversity, all the ones that we just kind of discussed today. And the results are provided in percentiles. That's one thing that we forgot to mention early on. That middle row, that RHIZE score, those are presented in percentiles. So if you're in the 50th percentile, then you're on average based off of the samples that we've processed in the past. This is actually an example of what we can provide with our greenhouse testing. So what you'll see at the bottom, this yield prediction should actually be genetic potential. I like to say genetic potential instead of yield prediction, but what we have actually been able to do is correlate that first 1 to 2 week of propagation to end of the season yield. We have a really strong correlation with an R-square of between 85% and 92%. It seems to be getting better over time, but we'll run a control. And if you want to trial EnSoil on your soil and your seed, we can trial EnSoil with your soil and seed. We'll look at all of these different pathways. We'll look at how we'll see the improvements in, nutrient cycling or mycorrhizae availability or number of genera, all of these key insights. And then at the bottom, we'll give you a total score on the total health of that root system. And we actually, because we have a really strong correlation with yield, end of season yield, we'll provide you with an estimated bushels per acre. And you can actually trial, I know EnSoil, because it's a biostimulant, it pairs really well with different biologicals. It could be a carbon source, it could be multi-species compost tea, could be a humic acid, a fulvic acid. When you're dialing in your program for this year, this is just a really great way to fact check and make sure that everything seems to be going as planned, that you would expect to see out in the field. This is kind of some data. We have more data points on this, but right now we're at around 0.92 per R-squared, which is basically the accuracy of our ability to predict yield with that measurement. With the RHIZE score. This is our greenhouse. And this is the submittal sheet that you would just fill in information on how you would like us to be treating your soil and your seed with different products. And we've got algae fresh on the shelf. Typical turnaround time is 21 to 28 days. So it's 1 to 2 weeks for propagation and 1 to 2 weeks for DNA sequencing, extraction, and analysis. And that is it. Wanted to open up the floor to any questions.
Just want to thank you guys again, Josh and Keenan.
Yeah.
Awesome presentation. Feel
Thank you.
free to unmute yourselves or throw questions in the chat. We got about 15 minutes left.
Josh and Keenan, I have something I would love for y'all to talk about. Something that was most surprising and also enjoyable to learn through some of these tests was the way that we're describing the EnSoils and interacting in the system. Could you explain a little bit about the community structure, evenness, diversity, and how we were talking about EnSoil really being utilized by the plants for right recruitment and in the rhizosphere for right recruitment for nutrient assimilation?
You got it, Keenan.
Sure. Yeah, that's a great point. We ran a little fast through some of that. It's already a pretty long-winded presentation, so thanks for reminding us on that. But yeah, so I'd mentioned real quick that we look at a couple different metrics of kind of measuring who's there, right? And the conventional wisdom is typically more is better. And so you would think if there's a bunch more microbes present, they're going to be doing a better job, but that's not necessarily what we've seen in the data so far. And sometimes actually, certainly if you go and test, between the rows, right? If you just grab a bulk soil sample and you compare that to a root sample, there'll actually be fewer species of microbes in the roots than there are in the bulk soil. And so what that says is that the plant is kind of recruiting, it's gatekeeping a little bit and making sure that the right microbes are getting into that root zone. It doesn't need everything. It wants those, rhizobium, and it wants some of these species that can perform those key functions, whether it's, nitrogen, potassium, phosphorus cycling. And that's something that we have seen in these roots to shoots trials with EnSoil, is actually the maybe even a slight reduction in the number of species, or in the what we kind of some of the diversity and evenness metrics, but that actually corresponds with improvements in the microbial nutrient cycling potential. And so, like Meredith alluded to, we theorize that that's basically, recruiting the right microbes. And you can kind of think of, I think we've talked about before, is, you can have too many cooks in the kitchen, right? You don't want everybody trying to do the same things at the same time. You want the right microbes in the right place at the right time. And that is something that we seem to see from these trials, at least this year. And I'm very excited to see. I have a good feeling about it going forward, that we'll continue to see that as one of the effects that EnSoil seems to have. And it definitely seems to improve crop health overall, and that kind of nutrient uptake efficiency. Definitely, Any other thoughts, Josh?
No, I think you hit it right on the head. I think was it Dr. White? Dr. White described your product as basically acting as a cover crop around the root structure. Unfortunately, we don't do microscopy, though that might be something that we implement for future trials. But whenever I think of the EnSoil Algae product, I think of it as basically attaching the root system as a cover crop, and it's gatekeeping and guarding the root system and recruiting the right microbes to get into that system. So that's probably why we're seeing lower, number of genera. Not much. It might be a difference of, 200, 100, 200 different genera, but kicking out some of the microbial populations that might not be serving the population. Maybe they could be dead and they just taking up space, and so that EnSoil algae could just be moving and cycling and more efficiently creating an environment, like I mentioned before, working to balance the pH, working to help the crop with producing more carbon fixation, making more space for the right microbes and for the crop to manufacture and engineer more efficiently the microbiome that it needs to serve the specific functions that are required.
We got some more questions.
Steve's wondering, how and when the EnSoil was applied for these plots.
So it was overwhelmingly in furrow. I think there was one or two farmers that also did a side dress, but that was the idea was in furrow, and that's something that we tend to see. Like Josh mentioned, that kind of early stage microbiome is really important. The way you kind of set up that plant with the biology really early is going to have a strong effect over time. So we do like to see in furrow or some sort of 2x2 or something like that, a seed dressing application if possible. But we have seen some good results from later foliar applications as well, and all up in the floor. If anyone has any more thoughts on that. I guess
I think that's great.
If not, I'll jump in. The residual benefits. This is a really good question. I don't know about the crop residue. We have seen, I think, improved carbon and nitrogen breakdown, basically that breaking down that crop residue. So I wouldn't be surprised to see that it is acting over time. And that's one thing I think this year we're excited to get some kind of repeat trials and see how much we're building year over year, and how much of that biology is staying in the soil even when the roots aren't there. I'm very interested as well, because I would imagine that some of the kind of management techniques, right, the way that if you put any covers down, if there's any manure or anything like that going down, and whatever products you might use to burn down or control weeds, all of that's going to factor into, I think, how strongly those residual benefits are going to be able to show up year on year. But it's a very interesting question, and I'm excited to get some data on that this year.
Yeah, we'll be carrying these into year 2, and we also have some trials going on with total acres that will be year 2 in 2 different fields.
Yeah, if anyone's interested, we'd love to work with you. So Steve, great to see you or talk to you. You're asking, do we measure plant hormones and amino acid levels? So we have one biomarker. It's called plant stress adaptation, which is kind of a hodgepodge of different phytohormones like auxins, gibberellins, trihalus, all sorts of really important plant health promoters. And these hormones are responsible, for flowering or germination, helping to get a good plant resist pests and things of that nature. We will be actually breaking out those markers outside of just one score, and that'll be happening this year. But yes, we do look at plant hormones in the rhizosphere. The rhizosphere is really where a lot of that phytohormone production occurs. As it relates to amino acids, maybe Keenan, do you have any insight on that?
I was going to point out that it's important to note that with our technology currently, we're not actually measuring the plant hormones themselves. We're measuring the genetic capability of the microbes to produce them. And there is a strong correlation there, but we're not actually directly measuring the amount of, say, indole acetic acid or something that's in there. We're measuring how much can be made by the microbiome. And so for the amino acid levels, that's a really good question. We don't currently measure that. That's a different process than the metagenomic process that we do. But certainly something we're very interested in. And like I think I had mentioned, we do see there's a strong correlation with the organic carbon and nitrogen breakdown in amino acids. Breaking down some of those longer chain hydrocarbons will produce some amino acids, but we don't measure that directly. So that's a good question.
I'll call you, Josh.
All right, sounds good.
I think we have a couple more.
Hey, Trisha, it's been a minute. So you saw the Dr. White showing the rhizophagy on leaves enhanced with algae. What about foliar applications? Yes, I would say a good amount of nutrient cycling does occur in the leaves. We haven't done a lot of research in that area, but I think this year we might be looking at that a little bit more as it relates to different nutrient cycling pathways that are occurring in the leaves. So
we definitely do see changes in the soil microbiome in response to foliar applications. And so,
yes,
yeah, we're very interested to see kind of how that is working out. If signals are being transferred, if it's just carbon, or nutrients that are being moved, exudates. So there's definitely a relationship, and excited to get more data and see where that goes.
Yeah, can I add to that, guys? Hi,
Absolutely.
Trish. Definitely synchronistic in your asking, because just today I think we've identified a great researcher that Dr. White pointed us to do a study on algae as endophytes in the leaf zone and the phyllosphere. And guys, Keenan and Josh haven't even told you guys this yet, but we may add it to our to the Roots and Shoots program, add another to this for a field study, which is really exciting. We know there's a mechanism of action there. We see it enhancing other products that it's inputs that it's put out with. We have some white paper data on that, but we don't have it specifically on that mode of action with EnSoil. So we're looking to collect that this year and probably moving forward with that.
All right, sounds good. That would be exciting.
We got a great question by Steve as well, which is something that I maybe meant to bring up. About regarding if small amounts of, say, humics, fulvics, carbon, or diverse compost will act synergetically with EnSoil. And that is a really, both those humics, fulvics, carbon, that's a big one. I think Josh had mentioned it's kind of the food, right? It's kind of the currency or maybe of that microbial cycle. And we definitely see a strong response, probably a stronger response, if there's either really good soil carbon, if you have really high OM, or if you put down a carbon source, molasses, or humic, fulvic, something like that are really good because they can also have positive chemical effects on the chemistry of the soil system and provide that really key available carbon source to, you can kick the microbes in the pants and try to get them moving, but if they don't have enough food to chew on, they're not going to get very far, I guess. So yeah, carbon is a big one there. And diverse compost extracts, we've actually seen good responses for EnSoil along with like multi-species, compost teas, stuff like that. So it definitely can act. I think we've almost only seen it act synergistically. I can't say I've seen an antagonistic relationship that I'm aware of. So that's definitely a good question.
Yeah, and that is something that if y'all are interested in mixing and matching, we could trial in the greenhouse. On several occasions, we'll mix things like, organics or different worm casting products with algae, and there's always a positive response. In some cases, especially in soils that are kind of low on total organic carbon, typically there's a better response in those systems when you have more microbially available carbon.
Mm-hmm.
Awesome. Thanks, guys. We're almost at 8 o'clock here. So I'll put up our events. So we're going to have Josh and Keenan back a week from today for part 2, talking about beans and milo and maybe some more about the greenhouse stuff. So please join us for that. That link for registration will be in the follow-up email along with the recording for tonight. I know there were maybe a couple of questions we didn't get to, so if you reach out to us, we are happy to connect you and chat with you. We do have another webinar coming up the first week of April with the Minnesota Soil Health Coalition, and then 2 upcoming info sessions. So we would love to see you at any of the events. And Josh and Keenan, very grateful for you and our partnership and your info sharing tonight. So thanks for being here, and thanks to you all for joining us.
Thank you.
We'll see you guys next week.
All right.
Thank you, guys. There was a lot we didn't get to talk about, so all the guests here, please make sure you make it back to part 2.
I love how much we know about your product. We've done so much research on it. It's such a good product. So I'm really excited to work with y'all and learn more. Such an interesting, microbe. It's just Corella vulgaris.
Yeah, we're definitely these days describing it as a multitask and your everything tool.
Yeah.
Your favorite tool in the toolbox, everything tool. It's hitting a lot of notes. That was great, guys. Thank you so much.
Good.
Yeah, thank you.
Yeah, you guys rock.
Excited to get more data. I mean, imagine what we can learn over in a year's time and kind of as we get more data points on different growth stages in expanding out like the phytohormones. I think that's probably what I'm most interested in, is learning about like auxin production and things of that nature. So
how it relates to specific nutrient pathways and different things. There's a lot of cool data left to unlock.
So much we don't know. So much to discover. All right, everybody. Have a great night. We'll see you next week.
Thanks.